Okay, so this video is the AP bio version of Interphase. So we will talk about the cell cycle, G1, S, and G2, as well as discuss different checkpoints and chromatin. Okay, so in the cell cycle, this is basically interphase and M phase. And like I mentioned, this will focus on interphase. But what is the cell cycle, right? It's basically the steps a cell goes through throughout the course of its life. And when we say its life, like on its way to divide. And so when we think about, well, what's the big picture? Why do cells divide? Well, in actuality, all cells come from existing cells. So when we talk about a sperm fertilizes an egg, and we all start out as one single fertilized like cell, how we get to trillions of cells today is by mitosis. Our cells will divide and make identical copies. And you might be thinking, but I'm not just like a ball of cells that are all identical. And that is where gene expression and cell specialization comes into play, which is a whole nother discussion. Okay, so but why do our cells divide? Well, one, like I mentioned, is growth. But the other one is repair. So if you've ever had a cut, like your skin got cut, we need to repair that and build new cells. And that happens by mitosis. So basically, we start with one cell, and then it prepares during the cell cycle to divide into two identical daughter cells. And so what we are going to see is how all of the steps leading up to that actual division in this video. And then my next video will be M phase. Okay, so let's look at the cell cycle. So the cell cycle is made of two major parts. We have interphase here, and then we also have M phase. Now, interphase is 90% of the cell's life, and then M phase is actually where it divides by mitosis. Or if we're producing gametes, that'll be meiosis. Okay, so let's go ahead and see how we can divide interphase into three sections. So we have G1, S, and G2, and we'll talk about each of these in a bit of detail. So in G1, this is basically a cell carrying out its functions for life. So when we talk about, in my previous videos, we've talked about how like liver cells can store sugar as glycogen, like we'd be talking about a liver cell in G1 carrying out its functions, or like a beta cell in a pancreas producing insulin. Like it's living in G1, it's producing insulin, it's doing its job, it's carrying out its functions. So when we talk, I just mentioned how like all cells come from existing cells. So when a cell is first produced by mitosis, it's in G1, but it's a brand new, like just formed cell. So it will grow a bit and then carry out its life. Now, some cells will spend their whole lives in G1 and will never actually divide. So if a cell never divides, we call that cell, it's like arrested in G1, like it's not going to ever move past that. So it is, like we talk about it basically that it like will leave the cell cycle and enter into a state called G naught. And G naught is basically G with like a 0. So I always think about it as like G not dividing. So examples would be certain brain cells that, like your neurons, that aren't gonna divide. Liver cells, interestingly, will not, like they will leave the cell cycle and exist in G1 or G naught and stay there unless there's damage to liver cells. They can be like called back if they get a signal to divide and then go through the cell cycle. But other cells, like our skin cells, for example, only last for about 24 hours. So they're constantly dividing. They would not go through like this G naught phase. Like they would be in G1, then move into S phase, G2, M phase, and just keep on dividing. So the different lifespans of cells vary depending on the cell itself. So as I mentioned, the skin cells are about 24 hours. Your taste buds are actually specialized cells that are replaced every 2 to 3 days. This is why if you like burn your tongue and your taste buds are like damaged and you can't taste anything, eventually they fall off and you swallow them and new cells replace them. So your tongue's not burnt forever. Your red blood cells are about every 3 to 4 months are replaced. So anyway, our cells have different lifespans, and if the cells are going to divide, they're going to go through the cell cycle. But if a cell receives a signal to divide, and it causes it to like move past G1, it has to be checked first to make sure that all of the DNA inside of that cell is healthy, and this is a good cell that we should replicate. So there are these things called checkpoints throughout the cell cycle. There's 3 checkpoints that we will talk about within these 2 videos. And so our first checkpoint is the G1 checkpoint. And once a cell receives a ligand, like a growth factor, to divide and go past or go through the cell cycle, it's going to get checked at the G1 checkpoint for a couple things. First is, did it actually receive like a message to divide? Are there enough nutrients to support a cell dividing? But most importantly, is that DNA free from errors or damage or mistakes? So at the G1 checkpoint, any like mistakes or damage in that DNA can be repaired, which will be the topic of my video for like cell cycle regulation, which is in a few more videos coming up. But anyway, and if the damage is just too great, then the cell can go through apoptosis, which is programmed cell death, and will be prevented from dividing. Now, if a cell does like have damaged DNA or have errors or mutations and it ignores this checkpoint and it divides anyway, this is like uncontrolled cell growth, and that is basically leading to tumor growth. And if that tumor is malignant, well, then that would be like cancer. So these checkpoints are pretty important at regulating the cell cycle. So I think it's fascinating that once a cell moves past that G1 checkpoint, it's basically irreversibly committed to cell division, like it's going to divide. So if it's in G1 and it receives a signal, everything's checked out and it's good to go, and it leaves G1 and goes into S phase, it's going to divide. Okay, so now let's go ahead though and talk about S phase. So S is going to stand for synthesis. And synthesis means to make. So we want to think to ourselves, or what are we making in S phase? So remember, we're starting with one cell and we're hoping to end up with 2 identical daughter cells. So we need to take all of the DNA in the nucleus of that cell and make a copy of it. So each daughter cell has an exact copy of the original DNA from that parent cell. So the cell we start with is called the parent cell, and then it divides into 2 daughter cells, but they're going to be exact copies of each other. So in S phase, the cell synthesizes or makes more DNA in its nucleus. So here I have 3 strands of DNA, 3 chromosomes. In reality, in humans, we would have 46. And during S phase, this is where DNA replication happens. So DNA replication is its own beautiful process. It'll have its own beautiful video. But basically, you're starting with a double helix, and you make an exact copy of it. And so here, when you see this like X shape, these are like 2 like strands of DNA that are exact copies of each other. The only reason they wouldn't be exact is if there were mutations. So when we talk about mutations, these are basically mistakes that happen when DNA is replicated. So mutations would happen here in S phase. So anyway, let's study these little X-shaped things a little bit more. So here we have one single chromosome. So in eukaryotes, we have linear chromosomes. So like I have like here, like this string on my sweatshirt, right? So like we have linear chromosomes, which is different than prokaryotes that have a circular chromosome. So we have 46 like strings basically. And when we replicate or duplicate our DNA, we keep them attached. So this is one single chromosome here, but when we make a copy of it during S phase and it's still attached, this is still considered to be a single chromosome. However, it's called a duplicated chromosome. So here, this is one duplicated chromosome made during S phase. So a duplicated chromosome is made out of 2 sister chromatids. So if I use my hands, for example, like this is a sister chromatid and this is a sister chromatid, and they are held together at a region called the centromere. The centromere is where the chromosomes, or the sister chromatids, sorry, are highly condensed and attached. So they're actually held together all the way until mid-M phase, well, they'll eventually be separated apart. Now, in this centromere region is where we find like a cluster of proteins called the kinetochore. And the kinetochore, later we're going to see spindle fibers are going to come and attach to the kinetochore, and that's how we're going to like move our chromosome during mitosis or M phase. So again, let's summarize this slide. So here we have a single chromosome that gets replicated during S phase to make an identical copy. Here, this is still considered a single chromosome, but now it has twice as much DNA. So here, this is a duplicated chromosome. These are exact copies of each other. We call them sister chromatids. The sister chromatids are exact replicas, unless mutations happened, and they're held together at a region called the centromere. And in the centromere, we find the kinetochore. And the kinetochore is a group of proteins where the spindle fibers will attach. Okay, so sometimes we might see pictures of chromosomes. So like here would be a single chromosome, and then this would be a duplicated chromosome made of two sister chromatids. Okay, so let's go ahead and look at our nucleus a little bit more here. So here I have one single uncondensed chromosome. Remember, it's like a linear chromosome in the nucleus. Now, in reality, as eukaryotes, as humans, we have 46 of these. So like literally, I copy and pasted 45 times. So there are 46 chromosomes in this nucleus. Now, that's in G1, the DNA, it kind of looks like a bowl of spaghetti, like it's uncondensed DNA, and we call this uncondensed DNA chromatin. So chromatin is how our DNA looks in G1 and G2 and S phase. But anyway, so let's study a little bit more about chromatin. So chromatin is made of two things. We have these histone proteins and we have DNA. So in the nucleus, our DNA is wrapped around histone proteins. And together, that is what makes up chromatin. So chromatin in the nucleus is basically DNA wrapped around histone proteins. So therefore, chromosomes are made out of DNA and proteins. Okay, we will talk more about chromatin in gene expression in Unit 6 if you're in AP Bio. Okay, so how tightly we wrap the chromatin around the DNA will determine if our DNA is like loose and uncondensed or if it's like really tightly coiled up in a condensed form. Okay, so let's go back to S phase. So in S phase, remember that stands for synthesis. So here I have 46 uncondensed DNA chromosomes. So our chromatin here is our 46 strands of DNA loosely wrapped or wrapped around histone proteins. Now during S phase, we double the amount of chromatin. We double the amount of DNA. That is so much DNA. It's kind of like messy. And if you imagine, if our goal is to take that nucleus and make an exact copy and then make sure that each daughter cell gets an exact replica, if you just take this chromatin and rip it in half, you're not going to have identical daughter cells. And that is why later in M phase, we actually will take this chromatin and tightly condense it into that like picture you see of chromosomes that look like an X. So in reality, if you tell someone, think about a chromosome or picture a chromosome in your head, a lot of times they're going to picture this X shape. But in reality, our DNA most of the time looks like this bowl of noodles. It looks like chromatin. It's only when the cell goes to divide that you actually see this like X shape in M phase. Okay, so let's go ahead and look at G2. So G2 happens right before M phase. So this is the last chance for the cell to prepare and get ready for division. So certain enzymes will become activated to help the cell move into M phase. You can imagine a lot's going to be going on in that cell in order to actually divide into two. So we'll activate enzymes, the cell will grow even more, it'll make more organelles. So basically finish up preparations for cell division. Okay, but now in G2, it's very important. There's also another checkpoint. We want to think, well, what did we do from G1S to G2? G1 living its life, then it gets a signal, it's time to divide, and it goes into S phase. And in S phase, that is where DNA replication happened, where all of our single chromosomes became duplicated chromosomes. And then during M phase, we will divide them so each cell gets a copy. So in G2, it's really important at this G2 checkpoint that we check, did the cell finish replication? Did all of the DNA get copied? Do we all have 46 duplicated chromosomes? Because if you move into M phase without checking that, then your daughter cells may not get all of the DNA they need for life. So at that G2 checkpoint, we'll make sure that all of replication has finished and that there's really not any damage in that DNA, that it's all good to divide and go into two daughter cells. So that is the G2 checkpoint. Okay, so to summarize, we have our interphase made of G1, S, and G2, and we have two checkpoints. We have the G1 checkpoint to try and help ensure that we only replicate healthy cells. Only healthy cells go through the cell cycle that have good intact DNA. And then we'll study in detail some more what happens at that checkpoint in a future video. We also have the G2 checkpoint to make sure that all the DNA was replicated in S phase and the cell is ready for mitosis and ready for division in M phase. Okay, so that is interphase, and my next video will be mitosis, all right, which is M phase. So good job. Oh, M phase also has a checkpoint, you notice. Okay, good.